Welcome class. In this particular session, we'll discuss about the steps in glycolysis in detail. So this glycolysis, it is the common step both in case of aerobic as well as the anaerobic respiration. So that's why I have considered this particular step separately without including under aerobic or anaerobic respiration. So this glycolysis, it is also called as EMP pathway. So this is dependent upon the discoverer. So there are three scientists called as Emgen, similarly similarly Parnas. So these are the three scientists. So who elucidated the steps of glycolysis. So hence it is also called as Emgen-Meyer pathway. So, or it is called as EMP pathway. That is the alternate name for the glycolysis. And this glycolysis is considered as a universal pathway that is occurring in case of biological system, both in case of aerobically and anaerobically respiring organisms. So this pathway is seen. So that's why it is considered as an universal pathway occurring in case of biological system. And it occurs in the cell cytoplasm. So cell cytoplasm is the site of reaction for the glycolysis, because the enzymes that are required for the glycolysis, they will be present in the cell cytoplasm. It may be a prokaryotic or the eukaryotic cell. So in every individual cell cytoplasm contains enzymes essential for glycolysis. And glycolysis can be defined as it is a process of conversion of one molecule of 6-carbon sugar, generally we'll consider it as glucose, into 2 molecules of 3-carbon keto acid. The 6-carbon will be split into 2 molecules of 3-carbon compound in case of glycolysis. And the 3-carbon compound is formed as keto acid such as pyruvic acid. And this process is ultimately termed as glycolysis. And this glycolysis, it is going to occur in the 10 different steps. So it was 10 different steps. So we'll discuss about the steps of glycolysis in detail one by one. So coming to the first step. So this first step, it is called as phosphorylation. So first step is phosphorylation. So we know what is meant by phosphorylation, that is addition of phosphate into any substrate, that is called as phosphorylation. So here in case of phosphorylation, what happens? The 6-carbon compound, that is glucose. So here we can see the 6 carbons, 1, 2, 3, 4, 5 as well as 6. So these are the 6 carbons of the glucose molecule, and this is the ring structure of the glucose molecule. So it is a pyranose ring structure of the glucose. So here the 6-carbon glucose molecule will be added with the molecule of a phosphate. So this phosphate will be donated by the ATP. So ATP, it undergoes dephosphorylation, giving rise to ADP as well as the inorganic phosphate. And this inorganic phosphate that is released, so it will be added to the glucose molecule, and it results in the formation of a compound called as glucose 6-phosphate. So here it is called glucose 6-phosphate because, so here the phosphate molecule, inorganic phosphate that is obtained from the hydrolysis of ATP. So it will be added to the 6th carbon of the glucose. So starting from here, if we take a count, so this will be the 6th carbon, and to the 6th carbon the phosphate will be added, and hence the resultant compound formed is the glucose 6-phosphate, where this number 6 represents the number of carbon atom to which the phosphate molecule is attached. And this entire process, it is catalyzed by the enzyme called as hexokinase, and it requires magnesium ions for its particular action. So magnesium ion acts as a enzyme activator. So in the presence of hexokinase, so enzyme name, kinase is the enzyme which regulates phosphorylation and dephosphorylation process, whereas it is called hexokinase because, so here phosphorylation is occurring in case of 6-carbon sugar, that is glucose, or to the hexose sugar. Hence, the name of enzyme is hexokinase. So, in the presence of hexokinase, glucose will be converted into glucose 6-phosphate, and the phosphate for the glucose will be added upon hydrolysis of ATP into ADP as well as inorganic phosphate. Then, so followed by this, the second step, it is called as isomerization. So we all know that what is meant by isomerization. So it is a phenomenon which involves conversion of one molecule into its isomeric form. So it may be a structural isomer, functional isomer as such. So here, in case of glucose, if we consider, so glucose is a hexose sugar which contains aldehyde as a functional group. So here you can see the presence of CHO. So aldehyde is the functional group in case of the glucose, and similar to this hexose sugar, that is glucose, there is one more hexose sugar that is called as fructose, so which will be having ketone as a functional group. But if you look into the molecular formula, so it will be same, that is C6H12O6 will be the molecular formula. They have a same molecular formula, but they differ in terms of their functional groups here. So glucose is having aldehyde as a functional group, fructose is having ketone as a functional group. And this glucose and fructose, these are considered as a functional isomer of each other. And in the step of isomerization, so this glucose 6-phosphate, it will be isomerized into fructose 6-phosphate. That is, aldehyde group will be replaced by the keto group here, and this particular reaction, it is catalyzed by the enzyme phosphoglucoisomerase, or we can also consider it as hexoisomerase. That is the enzyme which catalyzes this particular reaction. So that is the second step in case of glycolysis. So followed by the isomerization. So the third step is again phosphorylation, where the phosphate will be added to that of the molecule to the fructose 6-phosphate once again. And here the donor of phosphate will be ATP. So here in the third step, the ATP undergoes a molecule of ATP. It will be hydrolyzed to release ADP as well as the inorganic phosphate, and this inorganic phosphate will be added to the fructose 6-phosphate molecule, resulting in the formation of fructose 1,6-bisphosphate. So this indicates that the phosphate that is donated by the ATP, it will be added to the first carbon of the fructose 6-phosphate molecule. As a result, both in the first carbon as well as in the sixth carbon, fructose will be attached with the phosphate. Hence, the name of the product is fructose 1,6-bisphosphate. And this reaction, it is catalyzed by the enzyme phosphofructokinase. And this particular phosphofructokinase, it is one of the important enzyme in the entire glycolysis, because so this absence of this enzyme, it may inhibit all the further reactions of the glycolysis. So it is one of the key enzyme in case of in the process of glycolysis. Suppose if enzyme is absent or if it is deficient in the body of individual, so the glycolysis cannot proceed further. So that's why it is one of the key enzyme, and it is also involved in the feedback mechanism. That is, when the more number of glucose molecules are about to oxidize, or they are undergoing the process of glycolysis, and the energy released is already sufficient for the cellular functioning. So this phosphofructokinase, so it inhibits the activity of the hexokinase enzyme. So thus the process of glycolysis will be stopped. Okay, so hence it is also involved in the feedback mechanism. So concentration of this enzyme decides whether the glycolysis should proceed or it has to be stopped. So that is regarding phosphofructokinase. So here the fructose will be 6-phosphate will be converted into fructose 1,6-bisphosphate in the presence of phosphofructokinase enzyme, and phosphate for this particular reaction, it will be donated by the ATP, which undergoes hydrolysis to release ADP as well as the inorganic phosphate. And the fourth step in the reaction is called as lysis. So it is the one step where exactly the 6-carbon sugar, it is broken down into 3-carbon sugars here, which ultimately gives rise to a 3-carbon acid as per the definition of glycolysis. So what happens in this step, we'll see. So in this fourth step, so fructose 1,6-bisphosphate, it will be split into a molecule of 2 molecules of 3-carbon sugars, such as one is called as dihydroxyacetone phosphate and another one is the glyceraldehyde 3-phosphate. So these two triose sugars, so these are again the isomers of each other. As we have glucose and fructose as a functional isomers, similarly dihydroxyacetone and glyceraldehyde 3-phosphate, both these two are functional isomers of each other. So glyceraldehyde 3-phosphate is the aldose sugar, whereas dihydroxyacetone phosphate is the ketose sugar. So this fructose 1,6-bisphosphate in the presence of water molecule and by the enzyme, so in the presence of enzyme, fructose bisphosphate aldolase, or we can consider it as simply as aldolase, which will split the 6-carbon compound into 3-carbon sugars respectively. So the upper 3-carbon molecule, they form the dihydroxyacetone phosphate, whereas the lower 3-carbon molecules or the 3-carbon atoms, they result in the formation of glyceraldehyde 3-phosphate. And these two are again the isomers of each other. So as a result, the fifth step is again is the isomerization. So in this step, so why this step is happening here? Because so there is no enzyme that can further utilize the dihydroxyacetone phosphate in our body. So that's why in order to continue the process of glycolysis, so this dihydroxyacetone phosphate, it will be isomerized into its functional isomer, that is glyceraldehyde 3-phosphate. And this particular reaction, it is catalyzed by the enzyme triose phosphate isomerase. So this triose phosphate isomerase, so it can catalyze the conversion of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate and vice versa. But in this reaction, as there are no enzymes that can further catalyze this dihydroxyacetone phosphate, so hence here in glycolysis, the dihydroxyacetone phosphate formed, it will be further again converted into glyceraldehyde 3-phosphate. So as a result, from one molecule of glucose, in this particular step, we'll get 2 molecules of glyceraldehyde 3-phosphate. And once 2 molecules of glyceraldehyde 3-phosphate is formed, so further reactions that are occurring in the glycolysis, it operates for 2 times because each reaction should occur twice as we have 2 molecules of glyceraldehyde 3-phosphate. So that is regarding the fifth step. So the sixth step, it is called as dehydrogenation. So the sixth step is dehydrogenation. So dehydrogenation means the hydrogen atom will be removed from the reducing agent or from the respective substrate. Okay, so that process is called as dehydrogenation. So here the hydrogen ion will be removed, atom will be removed from the glyceraldehyde 3-phosphate, resulting in the formation of a acid. Okay, so here glyceraldehyde 3-phosphate will be oxidized into glyceric acid, and the hydrogen ions released during this process, it will be accepted by the reducing agent called as NAD. So NAD, the full form of NAD refers to nicotine amide dinucleotide. So the full form of NAD is nicotinamide adenine dinucleotide. Sorry, nicotinamide adenine dinucleotide. That is the full form of NAD. So this NAD, it acts as a reducing agent here. So hydrogen ion that is released upon dehydrogenation of glyceraldehyde 3-phosphate will be accepted by the NAD molecule, and it gets reduced into NADH and the H plus, Because here the NAD plus, it can accept only one hydrogen at a time. In this process, two hydrogen molecules are released, where one will be accepted by the NADH and another will be in the form of the hydrogen ion itself. And along with this reaction is also coupled with addition of one phosphate to the glyceraldehyde 3-phosphate. Okay, but here so far we have seen whenever phosphate is added to the substrate, ATP is donating the inorganic phosphate molecule. But in this particular reaction, we can see that ATP is not acting as a phosphate donor. Instead, any other phosphate rich compound, they act as a phosphate donor. So here the phosphate donor may be either GTP, that is guanine triphosphate, guanosine triphosphate, or it may be cytidine triphosphate, or it may be creatinine phosphate. So these are the some of the phosphate donors that are present in our cell. So secondary to the ATP. So these also act as the energy compounds in our body. So here addition of phosphate from the molecule other than the ATP, that is called as substrate level phosphorylation. So hence in the 6th step of the glycolysis, we can see substrate level phosphorylation. So here substrate level phosphorylation refers to, so this substrate level phosphorylation refers to addition of phosphate to the any compound from substrate other than ATP. So that is called as substrate level phosphorylation. So here in this step, ATP is not acting as a phosphate donor. Instead, it can be obtained from guanosine triphosphate or creatinine phosphate, so which act as a phosphate donor. So that's why it is an example for substrate level phosphorylation. So related to this, a NEET exam question can be asked. Where exactly in glycolysis you can see the substrate level phosphorylation? So it is seen during conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglyceric acid. Okay, so the step is called as dehydrogenation. So here, in case of glyceraldehyde 3-phosphate, it loses hydrogen ions which will be accepted by the NAD plus, and it gets reduced into NADH plus H plus. And in addition to that, a phosphate molecule will be added. So already one phosphate is present in the 3rd carbon, and one more phosphate will be added to the 1st carbon of the glyceric acid, resulting in the formation of 1,3-bisphosphoglycerate. And the reaction catalyzed by the enzyme called as glyceraldehyde 3-phosphate dehydrogenase. So this is the enzyme that catalyzes the conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglyceric acid. And once this 1,3-bisphosphoglyceric acid is formed, so the 7th step is called as dephosphorylation. So far we have seen the phosphate is added to the substrate. So here dephosphorylation occurs. So whereas in dephosphorylation, the phosphate present in the substrate, it will be removed and it will be added to the ADP to form ATP. So that is called as dephosphorylation. So in case of dephosphorylation, what happens? The phosphate present in the 1,3-bisphosphoglyceric acid, 2 molecules of phosphate will be there, out of which 1 molecule will be removed, and that phosphate removed will be accepted by the ADP to form a ATP, and the resultant product formed will be the 3-phosphoglycerate. And the reaction is catalyzed by the enzyme phosphoglycerate kinase. And once this 3-phosphoglycerate is formed, so now this position of the phosphate group will be changed in the phosphoglycerate molecule, where from the 3rd carbon, the phosphate will be shifted to the 2nd carbon, and it results in the formation of 2-phosphoglycerate. And this step, it is called as rearrangement. So where the phosphate group is rearranged in this particular step. From 3rd carbon, it will be shifted to the 2nd carbon. and the resulting product formed is 2-phosphoglycerate, and the enzyme catalyzing the reaction will be phosphoglycerate mutase. And this interconversion or rearrangement, it occurs in the presence of a water molecule. And then once the 2-phosphoglycerate is formed, so next the process occurring is called as dehydration. So dehydration means removal of a water molecule. So from the 2-phosphoglycerate, a molecule of water will be released in the presence of enzyme enolase, resulting in the formation of an acid that is called as phosphoenol pyruvate. So this phosphoenol pyruvate in the last step, so that is in the 10th step, it undergoes dephosphorylation once again. So resulting in conversion of phosphoenol pyruvate into pyruvic acid. And here the phosphate released from the phosphoenol pyruvate, it will be accepted by the ADP molecule in order to form the ATP. So hence the resultant end product of the glycolysis will be pyruvate. So as this reaction from glyceraldehyde 3-phosphate, as every reaction is occurring for the 2 times, so here we'll get 2 molecules of pyruvate at the end. Similarly, so here totally we'll get 4 molecules. Here 2 molecules of ATP and here 2 molecules of ATP. So we'll get 4 ATP molecules, 2 molecules of pyruvate. Similarly, so here one NADH molecule is formed for one glyceraldehyde 3-phosphate. As 2 molecules are involved, we'll get 2 molecules of NADH as well as the H plus molecule. So this is the end products of the glycolysis. So here, in case of ATP, even though 4 molecules of ATP is formed, so the net ATP production in the glycolysis will be only 2 molecules. That is because, so 2 molecules of ATP are utilized in the initial step of glycolysis. So if we deduct those 2, then we'll get totally 2 molecules of ATP at the end of glycolysis. So hence, the end products of glycolysis includes 2 molecules of ATP, 2 molecules of pyruvic acid, as well as 2 molecules of NADH plus H plus molecules. So that is regarding the detailed account of glycolysis. So in the next step, we'll see the mechanism of anaerobic respiration, which mainly deals with the ethyl alcohol fermentation as well as the lactic acid fermentation.